Dalton Akert, everybody.
Yeah, Dalton here, and he's going to be talking about decoding fNIRS neural responses and machine learning approach. All the best.
Thank you for that introduction. In case you missed it, I'm Dalton Akert. I'm a junior here at Purdue's Weldon School of Biomedical Engineering. Before we begin, I'd like to give a quick thank you to my research mentor, Professor Maureen Shader. Our lab is in the Department of Speech, Language, and Hearing Sciences, and we do a lot of fNIRS imaging for auditory studies. So let's just get into this. So my particular responsibility in the lab is I work with brain-computer interfacing. So a brain-computer interface is anytime you have a brain, you have a computer, and they're going to transfer information either one way or both ways. That's a brain-computer interface. Some exciting work that's been done pretty recently is a company called Neuralink, founded by Elon Musk, was able to use brain-computer interfacing to get monkeys to play Pong. Think about that. And then also, that's the actual monkeys too. Additionally, there's been some promising research in the field of vision reconstruction. So using brain-computer interfacing to repair the vision from people who have become blinded, which is very promising. And then the way that we interact with the brain in our lab is we use what is called functional near-infrared spectroscopy. This is the actual cap that we use in our lab. Each one of these sort of optodes that juts out from the cap is going to be either a light emitter or a light detector. It uses a similar imaging technique to something like fMRI, which you've probably heard of. And then we particularly like fNIRS because it's non-invasive, the cap is relatively comfortable. And then also it's silent, which is huge for auditory studies because that means we're going to get less artifacts or structures that appear to be in the data that are not actually representative of our data set. So, how does this work? So essentially we use these two different wavelengths. We shine these two different wavelengths in your head. And then one of these wavelengths will detect oxygen concentration, and the other one will detect carbon dioxide concentration. So this is important because as your neurons fire and become more metabolically active, you're going to see oxygen rush to the spot that the neurons are firing to resupply their energy, which allows us to indirectly, by detecting the oxygen concentration and the movement and flow of oxygen in your brain, we are able to indirectly determine which centers of your brain are most stimulated and most activated at different times. So this is a typical fNIRS amplitude graph that you would see in a lab. Some key things to note about this is that your y-axis is going to be your change in concentration. The concentration levels that we're dealing with are like on the micromolar scale, so a millimole of a millimole if you are familiar with chemistry. This dashed vertical line here at time 0 is going to represent the onset of the stimulus, so when whatever stimulus we want to test, this is when it is applied. And then some key things to note is that, I can't use this, your oxyhemoglobin channels, which are the red blood cells that are carrying oxygen, they're going to spike around time 5 to 8 seconds after the onset of the stimulus. And then about 1 or 2 seconds after that, you're going to see a corresponding inverse in the concentration of your deoxyhemoglobin, or your carbon dioxide carrying red blood cells. So, for this specific study, we used one subject. We tested him 2 times every day for a week, Monday through Friday, and we applied 2 different stimuli. We used a block design process. So, can we do anything about this? I can't switch them. Okay, we're good. Thank you. Okay, so we use a block design process, which means our first stimulus we applied, which is our target stimulus, the stimulus that we are really trying to be able to predict later on. We applied 5 seconds of speech as our target stimulus, and our control stimulus was 5 seconds of silence. So we are trying to, at some point, differentiate using our fNIRS evoked neural responses. We're trying to differentiate the speech from the silent stimulus. So, and then additionally, we included only data from the left and right planum temporali. It's sort of on the posterior end of your temporal lobe in your brain. And it's your auditory cortex center. It's going to comprehend anything that has to do with speech or sounds that are related to some sort of meaning. And then this spinning brain that we have on the screen is Each one of these points represents either a source or a detector that we use on our cap. And then this is just to help you visualize where exactly this data is coming from. And then the plane temporale is somewhere around here. Okay, so like I said, we use machine learning to actually do this prediction and classification in this particular lab. Machine learning is a way that we can feed a computer information, have it learn the different features and patterns in the data, and then we can actually use that to predict information about future data that we feed it without it ever having seen certain features. So this first technique that we use is logistic regression. It's not really important that you know exactly how logistic regression operates. If I was going to get into the nitty-gritty behind how each of these machine learning models operate, I would take the next hour. But all you need to know really is that it's simple. It uses one estimator. If we have an imaginary data set where we have a certain number of purples and greens and they fall into this distribution, logistic regression is going to draw a line across the data set that classifies the most amount of purple points as purple and green points as green. So if we introduce a new data point into this set and we've only told it its position in this XY axis or plane, and we say it was right here, it would predict that that's a purple point, and here would be a green point. So that's just a very simple overview of how logistic regression works. The second one is much more complicated. It's called extreme gradient boost. It uses a form of gradient descent to actually use a number of different estimators and assign weights to each of those estimators and assign more weight to the estimators that are better at predicting the data. Some key takeaways is just that it's a more robust and technically sound model for working with large amounts of data that represent different underlying systems. So for the actual, testing. So we use what is known as leave one group out cross-validation, which just means if you have 10 data samples, you would take 9 of those 10 data samples, you train your models using each of those 9 samples, and then you take this last sample that you left out, which is why it's called leave one group out, you test your model with that sample, and then you iterate through your data until each sample has been used as a test sample one time and been trained on 9 times. So for the actual results, here we go. Okay, so this is a graph that represents our model's prediction accuracy based on what sort of data we fed it. So if we feed it only the data that represents your oxygen-carrying red blood cells, we see that we achieve a prediction accuracy using logistic regression of about the same, about 70% using only half of our data set. But as you can probably guess, the oxyhemoglobin carries less information than the oxy and deoxy combined. So by only using the data passed through the oxyhemoglobin channels, we're able to double the speed at which we're able to develop models and train them. So for the future of this study, we will not be looking at both deoxy and oxy, just oxy. And then this is what we're able to achieve using all of our samples that we collected. But what is interesting to note is that they all, deoxy, oxy, and them combined, all converge around the same prediction accuracy, but in oxyhemoglobin channels will do so faster than either one. So this graph is an interesting graph that is meant to hammer home how our prediction accuracy correlates directly with the ability of our study to evoke an amplitude, evoke a response in the oxyhemoglobin concentrations. So this line right here again represents the onset of the stimulus. You can see around the 5 to 12 second range, we see the greatest prediction accuracy for our model, which again correlates with the greatest change from baseline in each of our oxy and deoxy channels. And then there's also a couple different factors we have to account for when we're doing any sort of blood-related testing. So there are things that can introduce additional noise to our data. Specifically, physiological factors are important for this study. And we call anything that is a physiological factor that introduces noise we do not care about, that is systemic noise. So we talked about oxyhemoglobin, that's what we're mostly measuring. But the other factors that we could account for are your respiration rate, your heart rate, your galvanic skin response, which we measure on your palms, and it's a measure of your skin's electrical conductivity. So you sweat more, you are able to better conduct electricity across the surface of your skin. And then there's a whole plethora of different blood-related physiological markers that we also need to account for. So photoplethysmography is a measure of your blood pressure, temperature, I'm sure you can guess. And then oxygen saturation is a measure of the baseline concentration of oxygen in the blood around your body. So if we leave out the deoxyhemoglobin and oxyhemoglobin, and we train models only based on the physiological factors, we're able to see which physiological factors are most important in helping us predict what stimulus we apply. So as you can see here, this is all using logistic regression. There is one physiological factor that stands out among the rest as being able to somewhat reliably help us predict the stimulus we applied based solely on the physiological data. So that was GSR, which again stands for galvanic skin response. And it is a measure of your skin surface electrical conductivity. It has a documented link to emotion. It rises during stressful tasks and then falls during resting tasks. It's important to know that stress does not necessarily indicate like cortical response, like stress that you think about. It also indicates attention, which is why we are able to pick it up in a lab setting that is not necessarily stress-inducing, but may require some amount of attention. So if we use everything that we learned and we train our models based on the oxyhemoglobin as well as each of the relevant physiological factors, we were able to see that if we use oxy as a baseline, our model that uses GSR as well as oxy is slightly worse at predicting the stimulus that we applied, about 75-ish% to 70, but it is much less variable. It's much more precise, which means, which indicates to us that we are achieving a better and more technically sound machine learning model. So, and if we compare these two models with the models that incorporate all of the other physiological data using logistic regression, we can see that GSR and oxy only are leaps and bounds ahead of any other physiological factor that can be incorporated. And then another interesting thing to note is that, like I said earlier, XGB is a more robust and technically sound model for demonstrating lots of complex data sets that indicate multiple different underlying structures. So one thing that's important to note is that although we reach a lower overall peak prediction accuracy using XGBoost as opposed to logistic regression, we can see that the incorporation of potentially irrelevant data in our model does not really affect its ability to learn and predict. So to me, that indicates when we are going to be modeling in the future large, complex neural systems, we want a model that is going to be able to handle potentially irrelevant data, especially if we don't know what data should be relevant and irrelevant. So the applications of this research is it's one step in developing reliable fNIRS-based BCIs, which have a number of benefits over other BCI technology. We identified GSR as a relevant cofactor to account for in our predictions, which to my knowledge has not been widely researched in the BCI fNIRS field. And then we also identified a couple other things, a couple other factors. So we identified which physiological factors did not seem to be relevant, and then also we identified which scenarios lean best towards which classifier. So logistic regression, if you know your model or your data is going to be relevant, logistic regression might be the move if it's also not complex. But if you have information that's going to be potentially irrelevant, but you just want to try to learn off of anyway, XGBoost or another more robust and technically sound model is what you're going to want to use. So the next steps. The first step is to denoise the data. A wise man once told me that a wise man once told him that the best way to denoise your data is just to get more data. So that's step one. Additionally, we can apply some pre-processing and cleaning techniques to the data to sort of artificially increase the amplitudes of the oxygen concentration changes that we get, which would help increase our relevant signal to irrelevant noise ratio. And then also we can target additional neural centers in your brain. So in this study, we targeted only the auditory cortex and then compared our ability to predict whether a stimulus was applied to that or no stimulus was applied at all. An interesting study that we could do in the future would be applying a stimulus to your auditory cortex and then one to your vision cortex to see the effects of using a stimulus versus a different stimulus compared to a stimulus versus no stimulus. So, additionally, we could also gather more GSR data. We, like I said, collected it from the palms, but also equally valid places to collect from are your feet and shoulders. So it might be interesting to try to account for as many GSR data points as we can. And then additionally, like I said, I firmly believe that the future of BCI interfacing is in XGBoost and other more complex weighted models. So I'd like to give a quick thank you to all my lab members for coming and also my family.
I actually have a couple questions.
Okay.
So as for your models themselves, do you think something like topological methods or using homology would be more effective?
I haven't researched much in that. What specifically do you mean by that? Like what specific?
Such as for your point picking, instead of like that would allow you to see specific features in your data instead of like. Do you think that would help like features-based data picking?
So we actually tried some features-based data picking. There's a mild amount of features-based data picking going on here. So when we apply the stimulus, we actually collect the data points and associate all of the data points from 5 seconds before and 30 seconds after. So we use that as sort of like a pseudo feature to then relate all of those 35 seconds to one specific stimulus. So we could look into a little bit more of like a topological plotting, like you said. But yeah. Does that answer your question?
Yeah, and then just one more.
Okay.
As for your denoising, do you think something like sliding window embedding would be more helpful?
We actually use a sliding window for the time-by-time decoding.
Oh, really?
Yeah, so if you notice the maximum amplitude achieved in the time-by-time decoding, I can actually go back. Right somewhere. There. This maximum amplitude corresponds to the this is actually a mean of all of the different models combined, but this maximum amplitude corresponds to the mean response that we get in our 10 samples data. So we actually use a sliding window machine learning method and then kind of simplify it down to put in a box plot.
Very good.
Great.
Thank you.
Thank you.
We have time for one more question.
Okay.
Great.
Good.
We have